being here. So before we get started, I just want to do maybe. Why is that the case? Okay, figured it out. Just before we get going this morning, sorry about the technical issues, I just want to do what is a very Australian custom, and that is to acknowledge the traditional owners of the land that we stand on and that we're gathered on here today. And that here at Penn State is the Susquehannock, and so we want to recognize that and also pay respect to their indigenous knowledges and their elders, past, present, and emerging. So it's kind of a pleasure for me to speak to this group because I don't have to define what a microbiome is. I don't have to talk about what a microbiota is. And we can all very much appreciate that we live on a microbial planet and that every ounce of air we breathe, every bit of soil we touch, every desk that we sit in is covered in a microbial layer. And that all of these obviously have very strong interactions. And this is no different for the human body. Every ounce and every surface of the human body is completely covered in microbes, so much so that greater than 50% of the total cells in the human body are indeed microbial. And while I don't have to find microbiota, I will give you my favorite microbiota stat, and that is that the microorganisms of the body weigh about 1.4 kilograms, or about 3.5 pounds, and that this is equivalent to your brain, okay? So we're thinking not about a little bit of mass in the body, we're actually thinking about a substantial amount of matter in the human body that's contributing to our physiology. And I think the even more staggering stat is that these microbes encode 99% of the genetic information that's within the human body, 99%. And that's incredibly important if we're talking about how we might cure disease or how we might think about disease origins. We have to think about the 99% of the human genetic material that's moldable and that's changeable, that we know we can manipulate if we're going to think about new solutions to disease. And so most people talk about a lot of key critical functions in the human body that are provided by these microorganisms, including food digestion, vitamin production, drug metabolism, and interactions with the immune system, like immune system development or infectious disease protection, or even the ability of these microbes to detoxify most of the toxins from the environment. And I could stand up here all day and talk about the different metabolic diseases and different systemic diseases that are linked to the microbiome. But most of these, I would say, that have been studied in depth today have been done in the gut. And so, you know, as we can all appreciate, there's a lot of other microbiomes elsewhere in the body that are not the gut. And so I often say that's one of the things my team works on is every microbiome except for the gut. There's a lot of really great gut research going on, but I think that there are a lot of other diseases to look at. And primarily my team has been focused on the oral microbiome. And this is related to impacts of ancient DNA, which I'll get into in a second. But I'll just talk specifically about the oral microbiome for a minute because I think it's important to orient those of you who don't work on the mouth why the oral microbiome might be very interesting. So there's about 200 species of bacteria anyway that live in your mouth in every single individual, but overall about 700 to 800 species have been identified in the human mouth. And so a subset of these, probably about 10%, are shared across hominids. Whereas a much larger percent are localized to your environment, to your family, or maybe even to what you've eaten that day or what you've touched or breathed in. And what's fascinating about the mouth is that it contains tons of different micro niches. So you have soft tissue, you have hard tissue, you have your palate, and each of those houses a different type of oral microbiome. So they're incredibly specific. You can actually tease apart what's a salivary microbiome and what's, for example, a tooth surface microbiome. And this is one of the samples that we work with predominantly. This is what we call dental calculus. This is calcified dental plaque. And so when you feel that slime on your teeth, that is that bacterial biofilm growing and multiplying in real time. Maybe you drank a sugary Coke or something and you can just feel that slime. Those are the bacteria that are associated with the hard surfaces of your mouth. And as you go to sleep at night, this calcifies into a rock-hard matrix that's similar to cement. Okay, so you have the microorganisms encased in a sort of bigger, hard, rough matrix. And this preserves incredibly well in the archaeological record, which we'll get into in a second. But I think what's fascinating about it is that it's different on every single tooth in the mouth. So it really matters as to which tooth you've sampled and which surface of the tooth you've sampled. We can actually track which tooth it comes from based on the sort of composition, which is amazing. And these are incredibly intricate biofilm families. And so we have 200 species, and this is an awesome paper published in PNAS by Mark Welch a couple years ago, where they used fluorescent microscopy, where they marked 12 different species. So here they're only showing a couple. And we can see incredibly very much layered organization, and it's a very robust organization. And these microbes are very much in coevolutionary interactions with each other that have been developed over very long periods. So what becomes important in looking at microbiomes, I think, in modern humans today, is to understand how and why they change. If we know they're linked to disease, we want to know why they become associated with disease. Why would they change? What are we doing in our daily lives that may support a disease model of the microbiome? These are the factors for my lab that are incredibly important in understanding. But what's important is not only understanding their change in a modern context, but also in an evolutionary context. Because we may have different microorganisms simply because our ancestors underwent different evolutionary processes, had different environments, different diets, different lifestyles, and all of that will factor into the types of microbes that we also have with us today. So understanding the changes today in a modern context, as well as changes in the past, become incredibly important in understanding the origins of why we have the particular microbes we have, and how and why those microbial communities might change in the future, especially in the face of disease. And so there's a couple different ways you can track the evolution of a microbiome. One of them is to do comparative studies across different hominid species. And so this was a study done quite a few years back, but it is still incredibly robust, even in modern studies that have looked at similar changes or similar differences. And that was to compare bonobos and chimpanzees to humans living in the same environment. And I actually really like this study because they sort of control for an environmental difference. These are humans living on the sort of chimpanzee reserve, right? So they're exposed to the same environmental connections. Obviously very different diets and very different evolutionary histories. But what we can see is that primates and humans, despite living in the same environment, have a very different microbiome. But it also, again, doesn't sort of control for these different dietary and evolutionary differences. So another way is to use humans that are practicing traditional lifestyles as proxies to understand how the human oral microbiome, for example, might have been different in the past. And so quite a few researchers, myself included, have used different hunter-gatherer populations that live around the world to understand, what this pre-industrialized microbiome may have looked like. And so this was a study published in PNAS comparing wild ape species and sort of hunter-gatherer populations from Venezuela and Malawi, or rural populations, if you will, to people living in the United States. And what we see is, again, sort of differences across those cultures, but what we also see is a steady decline in the amount of diversity in industrialized peoples. And so this suggests to us that at least people living in industrialized countries, that there's really been a process or something that's occurred in our evolutionary history that would have resulted in a very different microbial assemblage in industrialized countries compared to other populations. And so we start asking questions about, whether non-industrialized people ever looked like hunter-gatherer people from a microbiome perspective, or is it really that just most people from industrialized countries are really of European origin? Is it really that the European evolutionary history is just different, and that's why we sort of see these differences? And so in order to do that, you've got to use ancient DNA. You've got to be able to go back into time and actually study what the origins of the microbiome were in Europeans, okay? Or if you wanted to, for example, understand the origin of the microbiome in Venezuelans or hunter-gatherers in Venezuela, you would have to go back to their ancestors and look at their microbiome to understand that evolutionary story completely. So we've been using ancient DNA to do this. Ancient DNA is simply DNA that wasn't intended to be preserved, but still exists in a sort of archaeological or even sort of environmental context. And you can get it from a wide range of different samples. You can get it from soil or bones, feathers, hair, eggshells. And it's incredibly varied in its preservation. It depends on the sample type you're looking at, depends on where that sample has originated from. But we do know that ancient DNA is unlikely to survive more than 1 million years. And in fact, the oldest reputable genome that's ever been constructed comes from a horse in the Arctic. This was frozen in permafrost for pretty much its entire life. And that's about 680 or 700,000 years old. And so Jurassic Park is not real, but at least we have a way of going back into time 1 million years. But it is quite difficult. I was mentioning that DNA degrades. And so there's quite a few considerations that you have to take into account if you're going to use ancient DNA in a context to understand modern processes or compare them to modern processes. So the first one is that DNA is damaged. It's really it starts fragmenting. So as soon as a cell dies, you get all these enzymes that are still active, and they'll start breaking down DNA, and they'll start sort of causing it to be in smaller and smaller pieces. And you also sort of get environmental chemical processes that can start to happen. So what you end up with is DNA that's very, very small. So to give you an example, in the Neanderthal microbiomes I'll talk about in a second, our average base pair length in one of those Neanderthals is 48 base pairs, okay? So we don't have to fragment our DNA, it's already pre-fragmented naturally. And the interesting part about it is, even though it's short, the sequence is also potentially wrong. And so some of these chemical reactions will actually change the base pair composition of our DNA. And so it's, you know, the wrong sequence and it's really fragmented. So imagine trying to put that piece back together from a metagenomic perspective, which we'll talk about in a little bit. But what's also fascinating about that damage is it also provides a signal for us to authenticate our DNA. And so there's been a couple programs that have taken advantage of this, bioinformatic programs, where we look at the sort of collection of thymines on the ends of the DNA fragments. Really what these are sort of conversion into uracils that happens during this degradation process. And so if we sort of see what they call a smiley face plot, if we see an enrichment to thymine at the 5' and 3' ends, we can actually say that that's an ancient DNA signal, okay? So if somebody is going to show you an ancient genome, always ask for this sort of signal and for them to show you that signal, because if it's indeed ancient, it should be there. But we also have issues with contamination. So modern DNA comes into our samples and can actually swamp out most of the ancient signal that we see. So we have to be incredibly careful with contaminants, both in the field and in where those samples are stored, as well as our own lab. We also have to worry about taphonomy. We know many of these species actually degrade at different rates. And so if you're trying to reconstruct the composition of a microbial community, you might have problems if you're going to try to look at abundance. You might have to sort of think about presence, absence. And on top of that, we have what we thought were some issues with reference sequences. You know, in some cases, we're dealing with evolution that's 100 or 200,000 years old. So if you're talking about a virus or a microbe, you may have differences in being able to map those sequences back to a genome successfully because there's been too much evolutionary divergence. So my team has been working a lot on how to sort of deal with these issues and deal with how we might technically and methodologically overcome these in order to be able to reconstruct an oral microbiome successfully. So we've been publishing quite a bit in this space. We've been looking at things like oral geography bias in the archaeological record in an oral microbiome. We've been looking at different ways to decontaminate ancient samples and sort of remove any modern contamination that might be present on them. We've been looking at methodology efficiencies, so improving our DNA extractions and improving our DNA library prep methods, which are already bizarre ancient DNA methods to begin with, you know, that take several days, but we're trying to improve those to get that out. We've been looking a lot at modern DNA contamination and trying to assess what those signals are in different labs and different kits, in different technicians that are processing your samples, because no reagents are free of microbial DNA contamination. People say that they don't have contamination. That's a false statement. We'll talk about this again. We've also been working on different ways to authenticate DNA. So we've developed a couple different bioinformatic programs that get away from reference-based mapping to authenticate ancient DNA sequences. And we've been working primarily with Daniel Houston at the University of Tübingen to improve ancient DNA species identification and being able to accurately reassemble these metagenomes. So just to plug for 1 second about contamination, because I'm talking to a microbiome group, which is super exciting for me. We have to be thinking about background microbial DNA contamination. And I think a lot of people are appreciating this. Where I still see critical issues with this today are mostly in the clinical field. A lot of doctors and clinicians aren't necessarily appreciative of the fact that there is microbial DNA in everything, and that none of your reagents, despite whatever company claims, if they're sterile, if they're DNA free, that just means the reagents have been gamma radiated. And that just means the DNA is there, but it's small and fragmented. So if you're looking at long amplicon sizes, it might not be a problem for you, but if you're looking at short shotgun metagenomic sequences like we are, it's a huge problem. And so we've done a lot to characterize these contaminant microbiomes, and we've published a couple different how-to papers on how you might deal with contamination. In this particular study that we published, it doesn't matter if you're dealing with an ancient or a modern microbiome. That's very different from this contaminant signal that you might see across different labs. But that contaminant signal is going to vary depending on whether or not you're in an ancient lab that has a ton of things to try to decrease contamination, or whether or not you're in a classical modern lab. It's also going to vary across the different reagents or different kits that you use. So instead of pretending like you don't have contamination, just ensure that you're monitoring it and trying to incorporate that using extraction-like controls and library application controls in your microbiome research. So back to the ancient DNA portion, we use a couple different sample types to go back into time and to reconstruct ancient microbiomes. And the first one that we use is actually coprolites, or preserved feces. I'm glad I'm not a coprolite archaeologist, because I think I would miss most of these in the field as rocks or something. But these are incredibly taphonomically biased. And so the second that this dung or feces is deposited on the ground, it immediately starts interacting with environmental microbes. And so you have this sort of increased environmental microbial content. And on top of that, the microbes still live for quite a while in that feces after it's deposited. And so you'll get growth of certain microbes that do well in that new environment. And in fact, some of the coprolites that have been examined today, they used, for example, mummies, and actually went inside of a mummy and pulled out some of the gut contents. And if you look at the microbiome profile of that, it actually looks more similar to a compost pile than it does to the human gut. And that's just because there's so much taphonomic change in that sample after the death of that person. So lucky for us, as I said before, we've been focusing on the oral microbiome, and here that's where you have calcified dental plaque. This is where we have that dental calculus. And for us, this has been a game changer in the ability to go back into time and look at the microbiome. So this matrix will preserve the oral microbiome, or at least that hard surface tooth oral microbiome, through time. And it preserves it incredibly well. It also preserves it back in the archaeological record for millions of years. So we can still find dental calculus on things like Australopithecines. So we're dealing with incredibly good, similar to bone type level of preservation in the record. What's also interesting about this, because it is rock hard, you can take it off the skeleton and you can actually soak it in bleach. And it won't disturb the signal inside, but it will help you reduce that contaminant signal on the outside. Okay? So it sounds crazy to take your sample and then put it in a vat of bleach, but that's what we do. And the idea there is to, and what we've shown actually is to remove a lot of that environmental DNA contamination that's on the outer surface. So we spent a lot of time going around the world and collecting different samples. We now have a library that's over 1,500 ancient calculus samples from around the world on 6 different continents. We've been spending a lot of time reconstructing different oral microbiomes through time. And we were able to publish the first sort of high throughput DNA study on this in 2013, Adler et al. Paper in Nature Genetics, where we showed that you could use amplicon studies to get some sort of signal from dental calculus through time. And we actually did some microscopy as well to show that it sort of even preserves the macro preservation in these samples. And this has sparked a whole new field in archaeology, really looking at paleomicrobiomics, which is a fun word to say. So since then, though, we've gone on to use shotgun sequencing now for primarily everything. So we're doing shotgun metagenomic sequencing for all of our samples. I'm going to tell you right now about the story that we're sort of seeing in Europe. But again, we've got the story done in South America now. We've worked on it in Africa quite a bit. And we have a new transect we're working on this in Asia and in China as we speak. So the story in Europe, what we did was go and look for very ancient human hunter-gatherers. But there aren't very many of them in Europe. They're sort of new to that area around 30 to 40,000 years ago. They're trickling in from Africa as the working hypothesis right now. What is present in Europe around that time, though, in much higher numbers, although they're dwindling, are Neanderthals. And so Neanderthals are an excellent proxy for what ancient human hunter-gatherers would have been like in Europe in that environment. They're well adapted to the European environment. And they would be sort of a great way of us to go back in time and look at what an ancient, at least hominid oral microbiome would have been like. We then just briefly at the beginning compared these to modern humans in Adelaide, Australia, as well as chimpanzees that were wild caught. And it's important that they were wild caught and not zoo chimpanzees. And what you can appreciate straight away, although this was with very low sample numbers, but it was more of a proof of concept study, what you can appreciate straight away is that Neanderthals and chimpanzees actually look much more similar from a large scale microbial composition compared to modern humans. And for me, what's frightening about this is that many of the taxa that are shown here in red are gram-negative, and many of them have been further identified to be oral pathogens. And so we do see an increased sort of immunoinflammatory microbial profile in modern humans compared to ancient humans. And I usually tell people, that if I could go back and do anything or give modern humans any sort of new microbiome, what I would do is go back in time and give them a Neanderthal microbiome. The Neanderthal teeth are incredibly, they're of incredible health. So a Neanderthal oral health record is, almost never shows periodontal disease. Cavities are very, very rare. And even when they are seen, they're seen in locations where Neanderthals were seen to be eating lots of carbohydrates, like acorns, for example, in the south of Spain. But Neanderthals have incredibly good oral health. And so we know that this is an oral microbiome associated with oral health. And we know today that this is probably an oral microbiome that's associated with disease. If we think about this from an evolutionary standpoint, it doesn't make sense to get caries and cavities and periodontal disease. If you were a wolf in the wild and you were getting that in your teeth and you were losing them, you'd be in big trouble. And you wouldn't be a very evolutionarily successful species. And if you think about many wild animals, they sort of do have good oral health, and they don't have the same sort of health diseases that we have in our mouths in industrialized populations. So that's kind of how we began to see the oral microbiome. So the way we took these Neanderthal oral microbiomes and compared them to a wide range of European farmers, hunter-gatherers, agriculturalists, and African pastoralists and hunter-gatherers to get a sense for how the oral microbiome may have changed through time. How do we get from this Neanderthal state all the way through to this modern human oral microbiome state? And the first big change that we saw was actually when people stopped foraging and they sort of moved into hunting and foraging. And so we see a switch in the microbiome, mostly a switch in the types of gram-positive sort of phyla that we see when people start hunting. And so we really think that the first major change in oral microbiome is something that occurs, 30-ish thousand years ago in Europe, and that it's something that's probably associated with increased meat consumption. The next major change we see is from hunting and foraging and that sort of traditional lifestyle into agriculture. So in Europe, this occurs about 7,500 years ago. This is when people really sort of completely change their lifestyle. So it's not just that people are eating lots of carbohydrates, although we think that's probably the primary driver of differences in the oral microbiome. It's also a time when people are domesticating animals, when they're not moving around as much, they're not nomadic, they're settling down, they're changing their sort of work lifestyle. And they're also doing things like metallurgy and exposing themselves to all sorts of new environmental hazards and interesting things. And then this is very different from what we see in sort of modern populations where we have this increase of gram-negative organisms all around. So we've come up with this hypothesis, which has been backed by a lot of other data. This is sort of initial stuff that we published in Nature, but it's been backed to show that hunting is the first major step of evolutionary change in the oral microbiome in Europe, followed by farming. And then this industrial evolution step is something that's occurred as well. And so we spent a lot of time looking at this change from the industrial revolution and trying to understand, how is the ancient agriculturalist different from a modern human? And we hypothesized it was something during the industrial revolution, so we've been working on that quite a bit. And it turns out that the industrial revolution does actually have a very significant impact on the human oral microbiome, in Europeans anyway. And interestingly enough, as we were doing that study, we also found another change that's happened sometime post-1950s. So we're calling this the Great Acceleration Change. It's something that happens post-World War II. Our best hypothesis right now is that that's actually probably linked to dental hygiene. It's also about the same time we start introducing very antimicrobial toothpaste into the human oral microbiome. So we know that we have quite a few different changes, at least in people of European descent, before we get to what we have today, okay? So we probably all started out with something that was similar to a forager-gatherer, something that was kind of shared across humans, Neanderthals, and chimpanzees, and that modern humans have gone through this sort of stepwise change to get to what we have today. But we can do things with dental calculus that aren't just reconstructing microbial communities. We can also do things where we reconstruct full microbial genomes and actually learn more just about bacterial evolution of an individual species. So this is an incredibly powerful approach if we want to just not think about microbiome research at all, but just think about pathogen evolution or even commensal evolution. And so one of the things we did in this paper was reconstruct an archaeal species that's common in ancient human mouths. It's called Methanobrevibacter oralis. This is a strain that we dubbed Neanderthalensis because it was found in Neanderthals. And this pushed back the oldest ancient microbial genome by about 46,000 years. So we sort of did a huge big leap and jump in this. But what's really fun about this particular study is it tells us something about what this species was doing in ancient humans and in ancient hominids. So we used beast dating to try to compare this strain that we find in Neanderthals to strains that we find in humans. And when you look at the phylogeny, what you see is that these two strains diverged about 120,000 years ago. Now, for us, that was a huge surprise because Neanderthals and humans, from their sort of genetic differences, are thought to have sort of separated about 700,000 years ago. So if these microbial species have really just directly co-evolved with the human body, they should have a split of about 700,000 years ago. Because they don't, this suggests that at least humans and Neanderthals somehow may have swapped these species, or at least come in contact of swapping these species about 120,000 years ago. Now, the news took this and ran with this as humans and Neanderthals are kissing, because this is also the exact same time that humans and Neanderthals are interbreeding in the Near East. Well, that's a lovely story, and I would like to think about that, and maybe we could get at that in some other way. We don't know that that's the case, right? They could just be in similar environments. They could be eating similar food sources. Maybe they could be parenting each other's children or something crazy as well. But it at least tells us something about the origin of these species as to when they come into the human body and how they might have come across in other species. So the studies we did in Europe straight away were very low resolved, and so we've also spent a lot of time collecting more and more samples, as I said before, but trying to improve our resolution of not just across all of Europe, but go into a single country and really reconstruct in great detail a more recent sort of evolutionary history of European oral microbiota. And this was a project done by one of my PhD students, Andrew Farr, who's now at the Cambridge Science Center as an outreach communicator. And he did sort of a British survey. He's very British, and so it was classical that he would do a British survey. But we did 172 individuals from Great Britain. And the first thing we did, which was a first for archaeology, was to show that this tooth type and surface really does impact your interpretations. And so if you're going through the archaeological record and you just find a random tooth, that may not actually be beneficial for your study unless you can sort of find the same tooth across different individuals, and that you need to sort of account for this oral geographic signal that we see in an individual across your different types. But with this data and with some biases controlled for, we could then do some really creative things about how the British oral microbiome has been evolving through time, mostly from about 4,000 years ago to the present. And what stood out for us straight away was actually something quite shocking, and that was that there was actually two groups, regardless of time period, regardless of location or culture. There were two groups of oral microbiota in ancient Britain. And what's fascinating about it is that group one was found a little bit more commonly towards the modern day, but not significantly so. And group two was a bit more present in ancient individuals. But we immediately started asking questions about why there were these two different groups. And group one is Streptococcus dominated. And so if you read in an oral dentistry textbook about oral microbiome today, you'll see that it's going to be dominated by Streptococcus species. Those are the species that bind the enamel of your tooth surface. So they're really fundamental to making the rest of that biofilm on the tooth surface. Group two, however, has a bit of Streptococci, but it's really not the dominant species. What is dominant is this archaea that we were looking at before, Methanobrevibacter. And it's Methanobrevibacter oralis, more specifically, but there are a couple other Methanobrevibacter species that come in and out. So we started really trying to understand why this change would have occurred, and we looked at it in much greater detail. And the first thing we did was actually to look at the microbial functions. Most of the large changes that happen in the human microbiome, at least in the gut, have been attributed to diet. And there's a lot of evidence coming out suggesting the oral microbiome is also sensitive to diet. It makes a lot of sense. You're chewing on food and it has to go through your mouth to get to your gut. And so we looked at these microbial functions as a proxy for potentially dietary functions. And indeed, you sort of see a beautiful signal that was almost too good to be true. So group one, this group that's dominated by Streptococci and looks like a modern oral microbiome, also contained a lot of functions that were incredibly similar to what we see in modern people, okay? So even though we're finding this in the archaeological record, it really is the precursor to the modern oral microbiome. It probably still goes through further changes that we see, during the Industrial Revolution and maybe even during the Great Acceleration. We still have yet to look at that. But we know that it exists, at least in the UK, about 2,000 years ago. What is fascinating, though, is that this group two that we looked at, that's dominated by Methanobrevibacter, actually doesn't contain functions that we see in many modern oral microbiomes. And what it's reminiscent of is actually a diet that's absent of sugar, absent of milk, and very high in fiber, and very high in plant material, okay? So we really think that this is probably a very wealthy dietary signal. These are people in the UK that are probably drinking milk. They probably have access to sugar when it first starts coming in from Asia and from the Pacific. So this is probably a very, very different microbiome type that's probably settled on diet, but I would be happy to discuss the million other reasons why we think that this could be the case as well. But one of the things we also did, because we were still interested, was to go in and actually look at any other skeletal markers that might tell us more about these sort of differences between these two groups. And what you can actually see is that the Methanobrevibacter community that's associated with that diet that's high in plants and low in all the rich things is also associated with skeletal signs of stress. And so again, it sort of, supports this idea that we're looking at a microbiome, at least these individuals, of people that are probably of low socioeconomic status, and people are probably doing a lot of labor and a lot of things that would also put a lot of stress on their bones. And so for an archaeological or for an anthropological context, this becomes incredibly useful, because now you might have an additional marker that can help you go back and look at socioeconomic status in the archaeological record, where you don't have that information otherwise. So this is all fine and dandy, but I told you that we were really interested in understanding this and the trajectory of the modern human oral microbiome. And so really, I want to get at the point that all these evolutionary changes really do have implications, and they do have a lot of importance for looking at modern health today. This evolutionary signal is not just something that's interesting scientifically, it actually matters for our health and how we think about health and treat health. And so what I want you to do is think about populations that are still hunter-gatherers today, and think about how that might be relating, or how that might relate to Neandertals and European hunter-gatherers that existed tens of thousands of years ago. They would probably share an oral microbiome that's much more similar than individuals who are living an industrialized lifestyle, or who had 7,000 years to adapt to agriculture and industry and even World War II. And because I was working in Australia, we used Aboriginal Australians as the proxy to look at sort of modern hunter-gatherers. A lot of these individuals were hunting and gathering until 150 years ago. And in fact, the last sort of Aboriginal Australian came out of the bush in 1956. And so it's a very recent process. It's not something that, you know, has been going on for hundreds of years. These people are still, in many cases, practicing active hunter-gathering, but were, you know, solely hunter-gatherers until 150 years ago. And what's fascinating about them from a health perspective is that many of the diseases that are directly linked to changes in the microbiome are also much more, or much higher levels in Aboriginal Australians than they are to Australians of European descent. Okay, so we have this, what might be a microbial or microbiome sort of mediated difference. And so we really wanted to try to correlate a lot of these changes that we might see in hunter-gatherer to industrial microbiomes to these sort of health effects that we might also see in this population. Because a lot of people have gone in and tried to control for socioeconomic status, all sorts of other factors, health factors, access to healthcare, et cetera. And there was actually a huge program in Australia called Closing the Gap that tried to do this. And despite all those efforts, despite improving all those metrics, they still didn't improve any of these health issues, which means there's some other underlying factor, there's some other thing that we haven't accounted for and haven't looked at. And we would make the argument that it's the microbiome. And so in fact, we've been working with the Australian Centre for Population Oral Health and primarily Lisa Jameson to characterize the Aboriginal Australian oral microbiome in adults and children and relate that to oral disease and systemic diseases that we might see associated with the microbiome. And indeed, when you look at Aboriginal Australians who are suffering from renal disease or cardiac disease and have periodontal disease, they have a very distinct oral microbiome compared to Caucasian Australians suffering from the same disease, okay? So despite controlling for those disease factors, they still have a very distinct oral microbiome. And in this study, we actually decided to try to control for some of those active hunter-gatherer lifestyles. We went and sampled Aboriginal Australians that were living industrialized lifestyles in cities, okay? So these are people who are eating a Western diet all the time. They're going to work every day. One of them is actually an AFL football player, so you know it's like an NFL football player. So, so these people are not living a traditional hunter-gatherer lifestyle, but despite that, they still have a very distinct oral microbiome. And what was fascinating for us is that if you look at some of these individuals and you try to characterize some of the species, all of the individuals from the Central Desert region actually had endomicrobiome in their mouth. Endomicrobiome, I wish the entomologists were here because they would know that this is a species that's primarily found in termites. This is not a species that has ever been reported in human oral microbiome anywhere in the world before, but it's reproducibly present in modern living Aboriginal Australians from the Central Desert. What's fascinating about that is if you live in the Central Desert, you actually eat termites as a source of protein on a regular basis. So despite these individuals living industrial lifestyles and not eating termites anymore, what we've done is some phylogenetic evidence to show that this is really something that was introduced probably 30 to 40,000 years ago in the Australian oral microbiome, and is something that has now become passed on from generation to generation. And so this is a very unique signal that's tied to Aboriginal Australians' connection to country, and it's something that could then play very interesting roles in disease and how we think about that disease. So this species is incredibly good at getting energy out of very nutrient-poor or cellulose or very fibrous-rich sort of material. And so if you're living in the Central Desert, you can imagine how having this microbe might actually be an adaptation. It might actually help you obtain more nutrients from what you're looking at. But if you're living in an industrialized lifestyle and you have a Western diet, it may not be an adaptation, it may be a maladaptation. And what's fascinating about this too is that we have to think about it, I think, in the context of a periodontal pocket and how its ability to interact with those other species may actually be influencing the etiology and the origin of periodontal disease in Aboriginal Australia. So we've been working with a lot of other Indigenous people around the world to do similar studies, with the idea being that everybody's going to have unique evolutionary stories, and that these different evolutionary histories and evolutionary stories will play into the types of microbes that they have, and that these types of microbes may underpin disease differently, and they may be unique ways of treating disease, or certainly other considerations that we may need to take into account when we're thinking about how we might cure disease, especially on a personalized medicine type scale. So I told you all about humans, and that's really fascinating, but I thought I would also do a plug for the fact that we don't just look at humans. We look at a wide range of microbiomes. As long as they're in an ancient context, we're game. And so the Neanderthal Oral Microbiome Project actually informed quite a bit of environmental research, which sounds bizarre, but maybe it won't sound so bizarre in a second. And that's because there was a lot of environmental ancient microbiomes that were being published. There's a ton of them that are coming out. And what's frustrating about them is they say they've taken their soil samples and they've dated them to a particular time period, and then they've sequenced the microorganisms present in that particular layer in soil, and then they say we've got 200,000-year-old microbes, we've got 43,000-year-old microbes, we've got whatever. The problem with it is that you also have living microbes that are in that soil sediment. And so unless you use sort of classic ancient DNA methodologies to go back and verify that those species are ancient and to verify that they actually are coming from the time period you expect, you indeed do not have an ancient microbiome. What you have is a mixture of modern oral microbiomes and maybe some ancient DNA in there as well, but you need to go back and suss that out. And so we really don't think that from a microbial perspective that an ancient environmental microbiome has been done yet. And so we used the Neanderthal oral microbiome project and the lessons we learned in that to go in and actually look at ancient environmental microbes. And we did this using a similar sample type. And so we went after calcium carbonate or calcite that was present in Antarctica. It was present in a location called Boggs. And what happens there is that a lot of the glaciers have retreated. And what you see in the ground surface after that are these big rocks that stick up out of the ground, and they're covered in about 3 centimeters of a calcite layer. And what that signifies is when that glacier was there, you've had water running underneath that glacier for long periods of time, just like you would dripping in a cave, and you sort of had the ability to build up what is the equivalent of a speleothem. But it's now, because of some of the glacial retreat in Antarctica, now sitting on the surface. And so we can go in and hit it with a hammer and take some of that calcite out, and then use similar methods that we used on the Neanderthal oral microbiome to dissolve calcium carbonate in the mouth with adenocalculus to get out the microbes and actually look at them and see what was there back in the past. And so these were actually some of the samples that we looked at. And we also did some sort of sediment controls and rock controls from that same location to really, again, verify that what we're seeing is not just a random contaminant signal or not something that's unique from that environment or from unique how we sampled it. And we, at this stage, used 16S RNA to look at this, but we've gone back and looked at shotgun since that time point. But this was, again, kind of early in the ballgame. And what was bizarre about these calcium carbonates is that they actually contained, using a lot of the geochemical evidence, they contained sulfate that was volcanic in origin, okay? We're in Antarctica, where it's ice covered, and it's cold, and it's been cold for a long, long time, right? So why do we have volcanic signals in this particular calcite? So we looked at the microbial signals that matched many of these layers to try to match up why we might have seen this sulfate and what sort of microorganisms may be underpinning that. And we sort of got out a nice microbial profile. It's actually very similar to other labs that have done a good job at looking at speleothems in a modern context. So it's a very similar microbial assemblage. And we were able to sort of control, using our other samples, what sort of microbiome you might see in that context. So we have a pretty clean ancient signal. What was very interesting to us, though, and what seemed to match with that sulfite signal was actually the presence of some thermophiles. Which again is a bizarre signal. So we didn't see this in any of the other environmental contexts that we looked at. We only saw that in this particular calcites. And so we were able to publish this in Nature Communications a couple of years ago, where we combined modeling, we combined geochemical evidence, we combined a whole heap of factors, ancient DNA, to come up with a working model about how melting and iron influx into the Southern Ocean may have sort of impacted some of our global climate cycles. And so basically, just to sneak you through it, what we think is happening is that we actually had ancient geothermal activity in Antarctica, and that sort of allowed for glacial melting. That's why we had this water running underneath the glaciers, and that's why you sort of have the buildup and the presence of that calcium carbonate. And that this sort of caused bioweathering of a lot of those rocks in the area, and this dumped iron into the ocean. And that iron dump was very, very significant for global climate changes because it caused a CO2 drawdown and actually had kind of large sort of global climate impacts. And so even though we can think about microbes at an ancient scale and we can learn from the Neanderthal oral microbiome, we can still come up with sort of very, very broadly important sort of models for how microbiomes might inform, again, things in our living world from an ancient context. So just to wrap up, I think ancient microbiome research reveals a lot about past people, and not only just their microbiome, but also their culture, their socioeconomic status, their health, all sorts of some fun things that we can use in the archaeological record. But I also think that we can translate that into modern living people, and we shouldn't make the assumption that all microbiome disease links and things we're looking at in an industrialized context actually translate to all cultures. They probably don't, and there's probably unique microbiome associations with disease in different human populations around the world. And last but not least, I think that understanding this evolutionary history and understanding how we get our microbiota, both in humans and the environment, really has the chance to have large impacts on how we think about the world today and how we might solve some of its biggest problems. So what's the history of your favorite microbiota? What's its evolutionary history? I want you to think about that as you leave today. And I want to think about what in their history might influence what you're trying to understand today. Is that an evolutionary signal you're looking at? Is there something that happened that caused them to have those microbes? And last but not least, what happened today that might alter the trajectory of that microbiome in the future? We're living in a very fast-moving world, and so what are we doing today that'll impact things in the future? So last thing is just a plug for the new Ancient DNA Lab. I'm heading up Penn State's new, very big ancient DNA facility. It's not just for us to do ancient DNA research; it's for anybody at Penn State to do ancient research. So if you have a project where you have historic samples, you have ancient samples, and you might be interested in looking at their microbiome in the past and integrating that into your research, please come talk to us. This is a lab that's open to anybody to use. There's obviously some training. Nicole in the back there, again, and will train people who are interested in doing their research in the lab. So please come talk to us if you want to do ancient DNA of anything. Even if it's fish species or something else that you're interested in, we can still do that. Doesn't have to be microbiomes either, although that's my favorite thing. And with that, thank you very much for listening and for deciding to come to this talk. Yeah.
So that was a really fascinating talk, and I have too many questions.
We can get a coffee later.
But I guess one thing that was really surprising to me was how each tooth has its own microbiome. And then I was hoping you could comment on linking that observation with how the diversity of the microbiome, oral microbiome, is decreasing as we start thinking of industrialized populations. And so are those microbiomes of each tooth converging, or are they still maintaining their own relative abundance for microbial species?
That's an awesome question. That's something we should do. We haven't done it yet, is the short answer. Because archaeological specimens are often quite difficult to come by, what we've been able to do is typically go after a molar from an ancient individual and sample the calculus from that. In good scenarios, we've gotten an incisor and a molar from ancient individuals. So in moderns, we can go all the way around the mouth and sample everything. But from the ancient record, because calculus is useful for a multitude of different other processes, not just DNA work, people can use it to look at phytoliths or plant remains or all sorts of things. There's even people doing isotopes now on calculus. It's been our strategy not to sort of remove all the calculus from some of these teeth because that's a very unique archaeological specimen. So we haven't looked at differences between the incisor and the molar as we go through time. And that's something that would be really, really interesting. And we have a study site in South America where we have multiple teeth sampled off individuals through time. And so that's something we could actually go in and really look at in great detail. We have quite a few different also traditional populations where we've looked at the whole mouth. The data is still coming though. We haven't sort of had time to analyze that, especially in the context of an archaeological records. So that's a really good question and something we should do. Yeah.
Amazing talk. One question in terms of the evolution of these different microbiomes. Did you look at coexistent patterns across your data set? Because since it's a very tight ecosystem, so many species can coexist and co-evolve.
Yeah, absolutely. So the oral microbiome is almost a model for that, right? So for example, when I talked about Britain and we see those two different microbiomes that are dominated by a particular species, there's actually a whole suite of species that come along with that. And the Streptococcus model is quite beautiful because that's what people have been working on for the last 60 years in oral microbiology, right? It's like Streptococcus bind to this species, bind to this species, bind to that species. And you can see those species all right there. It's beautiful. With the Methanobrevibacter dominate, there's a whole other set of species that come together. We don't know how those interact, and we don't know how those bind. Nobody's looked at that before because we didn't know it existed. And so there's a lot of work to be done, probably using genomic evidence and maybe some proteomic evidence to try to assemble those sort of microbiomes. We also have an in vitro model set up now where we can go in and try to recreate some of these ancient microbiomes, which sounds crazy, but we can go in and try to put in methano species and layer in these other taxa that we know coaggregate together through time, but we can actually then physically see how they bind with each other. That whole idea, though, is also incredibly important for thinking about how we might do oral microbiome transplants. So we know these species coaggregate together. We know they come together as a package. And certainly when we're looking at the difference between Neanderthals and chimpanzees, for example, and that hominid microbiome, it's missing one of these keystone species that allows for this coaggregation to come together. It's missing a lot of diversity in Fusobacterium and Leptotrichia species. And they're these species that combined the primary Streptococcus species and they combine the tertiary colonizers on the other side. They have both binding capacities. And so if you knock out that species, you then get sort of only this primary microbiome. And it's actually a very healthy, very small microbiome that doesn't build huge plaque and calculus structures. And so tons of people have been trying to target that species to knock it out of the microbiome. But because it's so efficient at binding tons of different things and it hides out the biofilm, it's incredibly difficult to target. But when we're trying to develop new microbiome strategies, we're thinking about how to eliminate that species or how we select for a co-aggregated group of species that might be stronger than something that includes that Fusobacterium. So it's an incredibly important point.
Your work with the Australians is like really cool. What is your hypothesis on how did this whatever termite thing stayed until today? Like how did it transfer through generations, even though they're eating completely different diets?
Yeah, it's really, really interesting. We are just now doing the shotgun sequencing on those Aboriginal samples to get a full genome of these oral endomicrobiome species. Because I would assume that there's some sort of adaptation that the endomicrobiome has gotten that allows it to survive within this biofilm, right? Because it has to get into the biofilm, but it also has to start coaggregating with these other species to sort of be passed down as a package. Because we, again, think this coaggregation is very important for which species get passed down, because they get passed down together. And so there's probably, my hypothesis is that there's some sort of genomic adaptation in those species that allow it to start coaggregating into this biofilm. And without that, you wouldn't see the sort of hereditary signal that we see with endomicrobiome. It's also very frustrating because what we've been trying to do is go back in the Aboriginal Australian sort of archaeological records. So I'm part of a Center of Excellence in Australia that's working to essentially write the history of Australia for the last 100,000 years because it hasn't existed. You know, if you look at history in Australia, it starts in 1850, right? Because there are written records before then. And so even at 1850, though, the early reports of Aboriginal Australians are of that having incredibly good oral health. So we wouldn't expect them to have really big plaque structures or really big, really big sort of coaggregation of species. And so it does raise a lot of questions as to how the species got in there and ended up being very successful. Is it really like a unique tertiary binder, that ends up being successful by chance? Or is it something that actually has some sort of unique function that becomes incredibly important in that? We're not exactly sure. Because right now it's in people with periodontal disease. And so there it's easy to imagine its function because it's in a huge, massive calculus where you could have lots of things binding in. There's lots of anaerobic pockets. There's all sorts of weird stuff going on. So you can imagine having lots of opportunity. But in healthy Aboriginal Australians, it's very difficult to imagine what sort of adaptations you would need to get that to maintain.
So your British study where there was the two groups, the Streptococcus dominated and the Brevibacter dominated. And so you sort of were suggesting that the two ones with Brevibacter were kind of like the lower class.
Maybe, yeah.
Perhaps.
Yeah, perhaps.
and you saw that their sort of like bone health and stuff wasn't as good as the other. What about their oral health?
Yeah, so we correlated that oral, that's a great question. So we correlated that oral microbiome to presence of caries and to presence of periodontal disease. And we didn't see anything significant. So it does correlate with dental abscesses. So we know that there is some sort of association with poor oral health as well. But an abscess is a pretty major thing, right? Like you've got to lose a tooth, you've got to have bone loss. In the archaeological record to see it, like it would have been a heck of a painful thing to go through. I would guess, though, that oral microbiome probably associates with periodontal disease, but we have some technical issues to overcome. And so the current ways of recording periodontal disease in the archaeological record, if you're doing biowork, and one of my students, Abby, will probably run into this, and she could probably fix all of it for us. But one of the issues is that there's different ways of classifying periodontal disease, and there's different ways of defining how drastic that periodontal disease is. And so in living people, you take a probe, a metal hook, and you stick it in between the tooth and the gum line, and however deep that probe goes in is how severe that disease is, right? But obviously there's no gum in people with periodontal disease, and so it's a bit difficult to describe. Certainly, you can try to see in some cases where the calculus is built up where the gum line was, if it's super gingival, and so that can help. But a lot of times too, you can also get tooth erosion from a lot of grinding and a lot of wearing on teeth, which is very common in ancient and historic populations. That has nothing to do with periodontal disease. It actually has to do with how the teeth are being used and what the physical activities are every day. And so I would say that the bioarchaeology, the assessment of the bones that's coming behind that information is probably not great. But we do know that it is associated with probably a poor oral health because it is significantly linked to abscess. There's a question in the back. Sorry.
It's not a question, just a comment. It's fantastic. A couple of days ago, I was doing a search. I work with microbes my entire life, and I was doing a search by myself to understand what's going on in the oral microbiome.
Oh, really?
And to understand my oral health.
Yeah.
And yeah, I'm glad to be here today. And I was surprised to know how many people are out there selling probiotics for oral microbiome. That's surprising.
Yeah.
It's a real business.
It is. It's a huge business, like $1 billion business. We were at the International Association of Dental Research Conference a couple of years ago in London, and people heard we were doing microbiome research, and it was like, come look at our probiotics. And so I don't know how many packets of probiotic chewable tablets I went home with. So they're like mints, like breath mints that you put in your mouth, but they have, you know, however many billion oral microbes. What's fascinating about it is the research suggests that you have decreased periodontal disease, for example, if you integrate these sort of probiotic products. And so we actually then worked on a study in Adelaide, Australia, where we looked at the introduction of lactobacillus probiotics into a mouse disease model of periodontal disease. Like, this is just completely crap, or is this actually meaningful in some way? And despite introducing a live inoculum, both through the mouth and through an injection into the gut, what you can see is that that probiotic actually does decrease the bone loss and periodontal disease in a mouse model. What you don't see, though, is the survival of that probiotic anywhere in the mouse, right? And so it's probably that there are proteins and things like that in the lactobacillus species that are, again, supporting the anti-inflammatory response that's so classically associated with lactose. But it's not that lactose is growing or doing anything in the mouth or in the gut, for that matter. We looked at both places. So they may actually be potentially beneficial if you had severe disease in some way, but it might make your breath smell better if it's got mint flavor. But you know, I think, yeah, obviously, just like the other probiotics, and I'm sure everybody in this room is kind of familiar with, you know, it's it's a bit of a sham in some ways. There's more question, yeah.
I had a lot of questions about the shotgun work you do and like how much of a sample you really need to get.
The shotgun. Sorry.
The work you do with, like, how much of a sample do you really need to get enough DNA out of it? Are there genomes for the different hosts that you're getting it from? I'm just curious about what that.
Yeah. I didn't talk about it at all today, which this is probably an audience we could have chatted about it quite a bit more. Usually people are like, oh yeah. So we actually use incredibly tiny samples. So in the archaeological record, obviously, these calculus samples are incredibly valuable. They're, you know, priceless. They're one of a kind. And so our general rule of thumb is that if you can pick it up with a pair of tweezers, you can use it. And so as long as you can soak it in that bleach and do that decontamination stuff, it's going to have, it will have sort of probably a signal. If the preservation is good, it will have that signal maintained. So we're dealing with incredibly tiny samples. And after you do DNA extraction, if you go to quantify your DNA using a qubit, you will see that it is 0. So these are the sort of levels of DNA we're dealing with. And we're, you know, you can kind of make it analogous to like single cell genomics. Like, it's incredibly low. And so we use ancient DNA extraction methods, which have shown to be 100 to 1,000 times more efficient at pulling out DNA out of samples. So they're not kits. And I'm happy to share those protocols that some of them published and whatnot. But it is an arduous process. It does take, you know, 2 days. It takes a day and a half in the ancient DNA lab to get that DNA out. You've got to do extra processes to improve that efficiency. But it is still a silica-based binding approach. We just use tons of silica, and we sort of do a gentler digestion at the beginning to make sure that we're not degrading DNA. The next step is the library prep. Again, we use a homemade library prep that's designed to work on short DNA fragments that are incredibly fragmented. But it's incredibly efficient. It's also very cheap. So we can do it for about $40 a sample, which is, you know, much cheaper than what people pay for kits. It does take about a day. So again, it's a bit longer, but again, incredibly efficient, and we can do lots of different things. We do double barcoding, and it's an Illumina TrueSeq type approach. Then for our shotgun analysis, what we've been using is a program called MALT. So I don't know if anybody's probably heard of it. It comes after Diamond, if anybody's heard of Diamond. So that was developed by Dennis as well. So when he won the U.S. Department of Defense $1 million prize for coding a good metagenomic program that could actually identify pathogens accurately, what they did was sort of develop Diamond and Malt during that process. And so it's about 10,000 times faster than BLAST. It's incredibly accurate. It does well on short reads DNA. What is annoying about it is that it needs tons and tons of space. So we have been running all of our analyses on a supercomputer in South Australia, which is the third largest supercomputer on the whole continent there. It's huge, and they have 1.5 terabytes of memory that we're running this on. So what you do is take all of your genomes of interest, and it creates a hash table of those and loads them up into memory, and then directly does the same thing for your sample, and then you're just essentially doing hash-table matching. So it's incredibly fast in that way. But to do that, you have to have lots of space because you have all of your reference genomes loaded up into RAM at the same time. And so we have been using a version of RefSeq of all of the full genomes that you can download from RefSeq at about 2017. I forget which month we downloaded it, but it's 65,000 bacterial and archaeal genomes. And so that's what we're using as our base. We've tried to go bigger than that, and you start blowing out memory. And so we've actually just been talking to Amazon Web Services. We can get up to 3 terabytes of RAM, which is insane. But it's, it's kind of one of the things we have to do to try to get, I think, the best analyses possible. And obviously, there's now a lot more reference databases. We've been working with Phil Hugenholtz, the Center for Ecogenomics in Queensland. And they've, have 120,000 really well-resolved sort of environmental genomes. And so we'd like to be able to tap into that sort of database and use that potentially rather than this RefSeq database that we have been using. But we are matching sequences to individual genomes. And we're also doing that in a competitive nature because we're doing it all at the same time. And so your cross-mapping is a lot less. What I think was also critical too is after we get done doing that, we put it into Megan and we use a least common ancestor, last common ancestor algorithm, and the LCA algorithm in Megan to help filter through those results too. Because with short DNA, you will have a lot of spurious mapping and cross-mapping just because things are happy mapping to multiple species. So that's a long-winded answer. A lot of, yeah, so that's kind of where we're doing. We've also tested other pipelines, so things like Metaflan also works well, Kraken works well. You're just not going to get the resolution out in your species diversity because we're dealing with environmental human kind of mixtures in a lot of cases. Databases, for example, that are biased to humans are going to be a problem for us.
Oh, thank you.
So I think we'll stop here, but maybe a talk on actually the technical aspects would be great in the future, right? I see a lot of, what can we learn from what you've learned?
Yeah,
might be
for sure.
really useful. And would you put back the questions that you posed to us? I would love to post those for us all to think about. Go for it. Thank you. And let's thank Laura for this great And it was recorded, so share it with others when it gets posted.